Hello everyone, I'm Brigitte Wimana from RTA in Uganda. I work as a molecular banana breeder. I'll be presenting on behalf of the breeders and the coordinator of Work Package 4. So this presentation was put together by Hannah Sheikh, the coordinator of the Work Package, and the breeders of different organizations contributed in their respective product. This slide shows the overview of the activities to be carried out by Work Package 4. In the first period, we started by putting together the set of knowledge of the different product profiles we're working on. In the second period, we looked at the different populations which could be having genetic variability required for genetic analysis for quality. And in the last period, we are working on understanding the architecture of these traits. But here we have to mention that for many products, we are supposed to be working with the tools generated by Work Package 3, but this has not been the case. So with the delay, we are trying to work with the traits already known by the breeders while we're waiting for the high throughput phenotyping method generated by Work Package 3. We'll be reporting on 3 activities. One of them is genetic architecture of users' preferred quality traits for variety, user, and socioeconomic environment in RTB breeding programs. And we'll be reporting on the developed genetic pools for quality traits for the breeding programs, and we'll be also talking about uploading the data we are generating on different databases. A number of trials have been set up in the third period of the project. For yam, 2 trials were set up in Guadeloupe by Synergies and Enecha, and 1 trial set up by Senecha in Côte d'Ivoire, and 4 trials were set up in Nigeria by ITA together with NRCRI or Mudike. For cassava, 1 trial was set up in Colombia by Alliance of Biodiversity and SEAT, and ITA together with NRCRI had 4 trials in Nigeria, and NACRI in Uganda had 2 trials. 1 trial was run by ITA on matoke, bananas, 2 trials by SIP, 3 trials by SIP on potatoes, and 1 on sweet potatoes. One general comment here is that the number of trials have reduced if you compare with the previous year, and this was one of the consequences of COVID-19, where the multiplication of the trials was slowed down. And because of the lockdown in many countries, the setup of the experiment was not done as planned. Breeders have been looking at correlations among the traits they've been working on. In cassava, at NACRI, they've been looking at the softness of cassava after steaming and boiling using 3 methods: sensory, water absorption, and penetrometer. And we see a negative, a very low correlation between sensory and water absorption, but a higher correlation between sensory and the penetrometer method, which is also positive. At the alliance between Biodiversity and SEAT, they looked at correlation between water absorption and cooking time, and this correlation was found to be negative and significant, ranging from 0.43 to 0.78. And in the same way, ITA and NRCRI or MODIC have been looking at the correlation between attributes in fufu and gari cassava. In yam, ITA looked at correlation among starch RVA properties in the material harvested from 3 locations. At Synergies, they looked at dry matter, protein, sugars, and hardness. And at Senecha, they looked at correlation among different quality traits, including fresh color, tube oxidation, dry matter, and so on. And in sweet potato, in SIP, they looked at correlation between optimum cooking time and other traits, and they found a negative correlation between firmness and beta amylase activity. And at IITA, we looked at correlation among sensory and other lab traits, and we found out that correlation among the sensory traits was quite high, which was as expected. And there was a negative moderate correlation between dry matter and the sensory traits. For heritability and genotype by environment effect, in cassava, they looked at two kinds of cassava, vitamin A content and the white fresh cassava from 4 environments. And they found that G by E was significant for the 2 types of cassava. And the same work was done also on fufu and the gari yield attributes in Nigeria. In yam, they found low to intermediate heritability for the RVA properties in cassava in 3 trials. And at Sihad, they found significant G by E, even though in some cases the main effect of environment was not significant. And heritability was ranging from 0.4 to 0.91. In matoke, the heritability of sensory trait ranged from 0.4 to 0.7. And for dry matter, the heritability was estimated as 0.68. Work has started on finding quantitative traits associated with quality traits in different products. Here we are reporting on QT analysis on dry matter and browning due to oxidation in yam as done by IITA. And also at CIRAD, they looked at QT associated with sugars and dry matter. And at SIP, they looked at beta amylase gene in 25 sweet potato genotypes, looking at its new variation in the region within exon 5 and 6. And remember, when we're looking at correlation, we saw that the activity of this gene is negatively correlated with firmness. A number of genotypes have been generated, evaluated for quality attributes and advanced. At NRCRI at Mudike, 14 yam genotypes were under trial at 4 locations. At Sihad, 7 new hybrids were selected and given to ENEHA for sanitization. And at Alliance of Biodiversity and SIAT, 12 clones out of 72 were selected for their low cyanide content. At IITA, together with NRCRI Mudike, 6 genotypes of cassava were found to be performing based on participatory assessment by the farmers, and 11 clones are now under evaluation in 6 districts in Uganda by NACRI. The phenotypic data generated by work package 4 are all going to different breeding bases, such as Musa base, cassava base, and sweet potato base. And in addition to the work done actually by work package 4, we have been providing material to work package 2 and work package 3 in the attempt to understand the quality traits at the lab level and also at developing the phenotyping tools. Meetings were held to boost synergies among institute and work packages. One of those meetings was for yam, gathering all the institutes working on the crop. Another one was for cassava, and there was a joint meeting for matoke, sweet potato, and potato. And the purpose of the meetings was for the breeders to sit in one gathering and have a discussion and share their concerns and experiences. The main issue that was identified in those meetings was that there was a lack of flow information between work package 2 and work package 4. Therefore, separate meetings were organized per crop between the 2 work packages. And one of the major decision that was taken in those meetings was that we should be collecting near spectra for all the breeding populations and also keep the flour for each sample so that whenever we have the phenotyping tools, we can use the samples for analysis. Also for matoke, a working team was put together to help with the analysis of near spectra, and this team is led by Fabrice Davrieux. in the coming period, we keep on having such meetings so that we can have the synergy flowing. In this fourth period, we are continuing with phenotyping the breeding populations using the methods that are available, the ones that the breeders have been using. We are also increasing the interaction with work package 2 and work package 3 in order to learn about the method they are developing and also be able to implement them whenever they will be ready. And at the same time, we're continuing to assess the architecture of the quality traits in terms of correlation among the traits, heritability, the effect of a genotype by environment, interaction, and also looking at the markers associated with the traits. We acknowledge the contribution of the different breeding teams led by the different breeders in the institutions working with RTBfoods, and also the different projects, Next Gen Cassava, GT4SP, Africa Yam, Sasha, and Accelerated Breeding of Better Bananas. This project is mainly funded by Bill and Melinda Gates Foundation, but Work Package 4 received some contribution from RTB Therapy and also from Bill, EU, GIZ, and USAID. Thank you for your attention.